Hello everyone, I'm Feixuan. Today I will talk about applications of DiLeu text for quantitative post-translational modification analysis and biomarker discovery. So first, some overview of PTM analysis. I will introduce the PTM and some different PTM enrichment strategies and some examples of its applications on glycosylation, phosphorylation, and hydroxyproline. So people used to think that one gene, one protein. However, now we know that it's no longer used for thought to work with, as one gene can make all sorts of different types of protein forms, and they exist from different translations, different single nucleotide polymorphisms, and the diversity of proteome increases through the post-translational modifications. Here is a example how the modification proteome and has effects on impact on the function. And these are all classes of protein forms of the same protein that affect their biological functions. For example, the phosphorylation, glycosylation, and hypervariable region may contribute to many disease progression. And that's why we need to study protein forms and especially consider their this modification, what they do to change what we think is a like basic gene product into some totally new functional moiety. So the whole point is PTM alter the function of the protein, and it can be a whole host of different ways. For example, we could change the shape of a protein by sticking a modification on it. We could change how quickly an enzyme acts on a substrate by putting a modification on it. We can also change how different proteins interact where they are localized in proteins in cells and all sorts of many other functions we want to understand. So this is the world of what PTMs do and what we might care about studying them. And these are just a few listed here, and I will go into in depth of a few of these. However, there are some challenges in PTM analysis. The first one is low abundance, like only 5% or less phosphorylations occurs in total proteins, and also poor ionization efficiency caused by the negatively charged ions such as sialylated glycopeptides, and also precious and limited samples may lose during the sample preparation such as desalting. So in order to remove non-modified peptides and make it easier for PTM to be ionized and fragmented, enrichment strategy is very necessary and required. So for example, the lectin is based on the recognition of the carbohydrates, and the helic enriches the glycopeptides through the hydrophilic properties imparted by the glycan. And bionic acids can covalently react with the cis-diols to form the 5 or 6-membered cyclic esters in basic conditions, and this reaction is reversible under acidic conditions. And in the following slides, I will show you some examples of applications of DiLeu text for quantitative PTM analysis using different enrichment strategies. The first project is a high-throughput simultaneous quantification of glycopeptides and phosphopeptides enabled by 12-plex DiLeu isobaric text and dual-functional titanium IMAC material. So some background about glycosylation and phosphorylation. So here is the figure of N and O glycosylation. So O-link glycosylation, sugars are added sequentially to the hydroxy-oxygen of serine or threonine residues on the protein, whereas the N-link glycosylation, preassembled blocks of sugars are transferred co-translationally via the amide group of asparagine residue. And glycosylation is also well known for its association with cancer progression. On the other hand, the phosphorylation state is mainly determined by the activity of kinases and phosphatase, and paper also showing that phosphorylation of fibronectin can lead to increased mechanical forces being needed for cell adhesion in various cancers. So our lab developed a dual-mode affinity approach for mono 6-phosphate glycopeptide enrichment. And later, our lab sort of expanded the titanium IMAG material to expand the hydrophility mode. And so the synergistic electrostatic and hydrophilic interaction can enable separation and fractionation of glycopeptides and phosphopeptides from complex biological samples. And further, our lab used the epoxy ATP titanium IMAG material, which is prepared by coupling ATP to a commercially available epoxy functionalized silica and then immobilizing titanium ions on it. And it will have a better, stronger hydrophilicity given by the hydrophilic adenine base and ribose sugar groups by the ATP. So this is the material I will use in my following experiments. So here is my experiment design. So proteins from cells will be extracted using detergent rapid-digest and sonication, and will be then digested with trypsin overnight at 37 Celsius, and peptide samples will be labeled with 12-plex DiLeu isobaric tags, followed by epoxy ATP titanium enrichment for glycopeptides in acidic condition and phosphopeptides in basic conditions. And later, the samples will run on Lumos, and the data is processed using the PD Bionic. And so here is the results of identification of glycosylation and phosphorylation in pancreas cells. So the labeled samples were combined in 1-to-1 ratios across all channels in figure A, and B is at 1 to 2 to 5 to 10 ratios. And we can see almost no phosphopeptides were eluted in the first 4 fractions, and only a small number of glycopeptides were eluted in the last 2 fractions, which demonstrate the good separation of these methods. And this showing the quantitative performance. So the DiLeu labeled glycopeptides from pancreas cell lysis were combined in 1-to-1 ratios or 1 to 2 to 5 ratio, and this is phosphopeptides combined in 1-to-1 ratio or 1 to 2 to 5 to 10 ratios. And these findings indicate this approach is suitable for the high throughput quantification of enriched peptides across a practical dynamic range. So now we have the evaluation and validation of the strategy, and we want to move on to more complex biological samples. So here I use the GDAC knockout mouse model. The GDAC is a crucial enzyme involved in deacetylation processes, and this is crucial for maturation and proper functioning of glycoproteins and other macromolecules. And patients with microdeletions that includes GDAC have intellectual disability and autism-like manifestations. So we hypothesize that in our GDAC knockout mouse, that decreased deacetylation of carboprotein would result in a decrease in glycosylation and phosphorylation, and hope we can discover the PTM signatures associated with GDAC and benefiting the detection of novel biomarkers and potential therapeutic targets. So here showing the results of glycosylation. At figure 1, we performed the hierarchical clustering of significantly changed quantified glycopeptide sites to explore their profiles in different groups. So the blue one is the knockout group, and the orange one is wild type groups. And we can see there is a decrease in glycosylation after knockout the GDAC. And for the so the figure B is the glycoprotein-glycan network diagram. So it maps which glycan modified which proteins, and we can see it displayed even distribution of the green one showing here the even distribution of high mannose and polyaminoes, whereas the fucose and complex species showed a higher appearances on multiple sites per protein. So these findings highlight the changes in specificity of individual glycosylations by defective transitions of the glycoproteins within the Golgi. And the figure C is the gene ontology analysis of biological processes, showed that the most significantly terms were associated with neuron development and function, representing the major biological processes of brain tissues. And moreover, we also discovered the glycan diversity and phosphorylation on the SHIPS1 protein. So this is tyrosine protein phosphatase non-receptor type substrate 1. And this is the transmembrane glycoprotein, which is crucial protein in the immune system that helps to regulate phagocytosis and immune responses. So here we successfully quantified 4 significantly changed unique glycoforms of SHIPs across 3 glycosides and identified a phosphopeptide with phosphorylation at site serine 409. And this site was not reported in the previous study. So our simultaneous quantification enrichment strategy enabled by DiLeu isobaric tags will facilitate more in-depth investigation of the cross-regulation of glycosylation or phosphorylation on these protein functions in future studies. So in conclusion, the study represents an effective analytical methods that enable simultaneous high-throughput quantification of glycosylation and phosphorylation. We also incorporated the one-tube sample processing workflow into the DiLeu labeling experiments, which not only simplify the experimental steps, but also greatly reduce the sample loss. Also, our results provide new insights into the GDAC-associated disease by indicating intimate involvement of this PTM. And future efforts will aim to expand the strategy to quantitatively enrich other glycosylation types such as O-glycosylation. So far, any questions? Okay. I will continue to the next.
The enrichment of phosphorylation only for serine and DiLeu or tyrosine as well?
Yeah, 3 of them will be enriched. But I think for IMAG, the phosphorylation for the tyrosine is lower. I think there are papers showing that before. Yeah. Yeah, it has different target. Yeah. So next project, boost DiLeu, enhanced DiLeu tagging strategy for comprehensive quantitative glycoproteomics analysis. So some background about the boosting concept. So the concept of boosting strategy or carrier strategy involves using additional samples to enhance the detection and quantification of low abundance peptides or proteins, as like showing here. And a separate high abundance sample is added to the analytical runs. So the purpose of adding the boosting channel is to enhance the signal of low abundance peptides by amplifying the precursor ion signals. And we can see that it can also enhance the peptide identifications and mitigate sample loss. And so far, these applications of boosting strategy found on single-cell proteomics, phosphopeptides in small population of cells, and not applied to intact glycopeptide analysis yet. So in 2022, our lab successfully developed a robust and highly sensitive strategy called DiLeu for enhanced isobaric labeled quantitative intact glycoproteomics analysis. DiLeu 118D is chosen as the boosting channel in the 12-plex experiment because it has no interference with other channels. So the overall sample processing workflow, so proteins from HeLa cells or CSF are extracted using RapidJet followed by trypsin digestion, and then samples labeled with DiLeu isobaric tags with boosting channel. And glycopeptides are enriched using helic material and further perform the high pH fractionation to improve the coverage. And then the samples will run on Lumos and using PD and Bionic for data analysis. So first, to validate the design of boost DiLeu strategy and evaluate the effects of different boosting channel to study channel ratios, B to S channels, in short, on the glycoproteomics coverage and quantifications. So the first channel in 12-plex DiLeu was set as the study channels, and the last channel, 118D, was selected as a boosting channel, and to generate different B to S ratios at 30, 50, and 100. And a control group was also prepared at the same time that only consists of the first three study channels. And the figure B shows that the similar reporting ion intensity distributions in study channels were observed in all groups, and none of the empty channels had an unusually higher reporting ion signal, indicating that 11D channel successfully served as a boosting channel and avoids isotope interference problems. And also the 30 boosting ratio is optimal in aspect of the identification numbers as shown here, and also the quantification accuracy. And after validation of the boost DiLeu methods, we further apply this on the human CSF samples. So in total, 18 aberrant N-glycopeptides in Alzheimer's disease CSF samples with site-specific information showing in the figure A. And this figure called weighted gene co-expression network analysis, showing 9 modules of glycopeptides, and 2 of them, the pink and red ones, were associated with AD phenotype. And gene ontology enrichment of these 2 modules uncovered these 2 modules was closely associated with brain development and extracellular function. So to wrap up this project, one tube sample processing workflow reduced sample loss and increased the glycoproteome coverage, and 30 boosting ratio is optimal in aspect of the identifications, quantification accuracy, and proof of principle experiment using CSF samples demonstrated the effectiveness of applying the developed strategy to size-limited biological samples, and represented the informative advantage of site-specific intact glycoproteomics analysis. And the third application is the proteome-wide investigation of proline hydroxylation in PDAC using the DiLeu isobaric labeling strategy. So some background about this disease. So pancreatic ductal adenocarcinoma is the 3rd largest cause of cancer-related deaths in the US, with a very low survival rate in 5 years, lower than 10%. And despite a lot of advanced treatment, the drug resistance of this disease also leads to extremely low and poor outcomes. And here is a figure showing how the ECM extracellular matrix changes during the pancreatic cancer progression. You can see they have increased ECM depositions and stiffness and increased osmolarity. So that indicating that ECM not only constitute a supporting issue, but also generate a specific microenvironment facilitating tumor growth, metastasis, and survival. And collagen is a major component of ECM, and in this project, I will focus on the specific PTM hydroxyproline on collagen and have deeper insights into the critical roles in pancreatic cancers. So as I said, collagen makes about 1/3 of the total protein content in human, and up to 40 of them carries hydroxyproline. So here is the triple helix is the main secondary structure of the collagen and consists of the repeated motifs glycine X or Y. And paper shows that altered activity of prolyl hydroxylases is associated with chronic pathophysiological conditions such as tissue fibrosis or cancer progression. So here is the experiment to analyze and enrich this PTM and also labeled with DiLeu labeled samples. So I used 6 pairs of malignant tumors and their normal adjacent tissues, and 6 pairs of benign tumors and their normal adjacent tissues. After protein extraction and digestion using trypsin lyase on the suspension trap, samples will be labeled with DiLeu isobaric tags, and the hydroxyproline peptides will be eluted with the modified helic enrichment. And samples are run on lumos and using Feixuan for data analysis. So first is the principal component analysis plot. So we can see the malignant and benign groups are well separated to each other. And also, I performed the hierarchical clustering of quantified hydroxyproline peptides. So the first 2 are benign groups, and the right 2 groups is the malignant groups. We can see the similar trends within each group. However, we can also see some more upregulated or downregulated hydroxyproline peptides in the malignant groups. And further, we look into the volcano plot to compare the comparison between benign tumors and their normal adjacent tissues, malignant and their normal adjacent tissues, and compare the malignant tumors and benign tumors individually. And we can see as the cancer progresses, there are also more and more significantly changed hydroxyproline peptides were found in each groups. And to look into the gene ontology analysis of the significant changed peptides, we can see the results of it shows the most significant terms associated with extracellular matrix and collagen organizations, and which is consistent with the studies on the impact of ECM dysfunctions in cancer progression. So far, this is the first study of hydroxyproline high-throughput quantitation in PDAC. And this dissolved-free strategy with modified helic enrichment is suitable for DiLeu-labeled hydroxyproline peptide analysis. And this comprehensive hydroxyproline analysis contributes to improved understanding of the critical roles of, that ECM plays throughout the pancreatic cancer development. So that's all for PTM analysis. Any questions? If not, I will let's welcome Haiyan to tell more stories about biomarker discovery. Thank you.
Hello everyone. For this part, I will talk more about the DiLeu as break tag in biomarker discovery. As we know that the biomarker is a measurable indicator of some biological state or condition. It also possibly a pharmacological response to a disease treatment. This biomarker is this biomarker play a very crucial role in several parts. Firstly, it can be helped in early detection of disease. Also, it can be monitoring the disease at different progression. Also, it can help to do the new drug development, and it also can be used to validate the treatment efficacy. Based on the role, biomarker can be classified into several types. For example, the diagnostic biomarker are used to identify the risk of disease, while the prognostic biomarker can provide information about the likely course or outcome of disease. Predictive biomarker indicate how well a patient is expected to respond to a specific treatment, while the risk biomarker can indicate the possibility of develop a disease for a healthy individual. While the safety biomarker can detect the potential adverse effects related to the treatment or intervention, while the response biomarker can help evaluate the effectiveness of the treatment, or maybe by a new drug, maybe by a kind of new method. Finally, monitoring the biomarker can track the change in the disease status or response to treatment over the time change. Each type of biomarker plays a role in many aspects of the disease management and therapeutic development. Biomarker can be obtained from many clinical sources, such as we can obtain the biomarker from the invasive sample, such as the tissue, blood, and CSF sample. We also can obtain the biomarker from the low-invasive sample, such as the saliva, urine, and exhaled breath. For the biomarker, if we want to see it be ideal, it should be meet some requirement. I think the first one should be able to quantifiable. The second one should be correlated to the clinical outcome, and it should be both sensitivity and specificity, and it also can be used to predict the disease progression, and also can maybe cost and effective and useful for personalized management. With the development of the technology, there are many types of omics approach can be used in the biomarker discovery, such as the transcriptomics, proteomics, and genomics and epigenomics, radiomics, as well as metabolomics. Among those omics technology, mass-based proteomics have been widely used in the biomarker discovery. This is play a very important role in the protein expression and study and protein post-translation modification, protein-protein interaction, as well as the spatial distribution information in diverse samples across the cell, tissue, and biofluid. Especially mass-based technology can simultaneously monitor the source of protein in a single sample. But to identify the biomarker, the technique must be high throughput, robust, and scalable to handle for the large sample size. Mass-based quantitative proteomics have proved to be highly effective and popular for lower biomarker discovery. Compared to the MS1-based quantitative biomarker discovery, MS2-based proteomics quantitative method, it can greatly enhance the high throughput, and it can decrease the run-to-run variability and greatly improve the quantitative accuracy. So it will be widely used in the biomarker discovery. Despite there have various MS2-based aspartic tag labeling technique such as TMT, ATRAC, and deuteric aspartic amine reactive tag, as well as our DiLeu, have been used in the biomarker discovery. But for this part, our focus will be on the application of DiLeu in the biomarker discovery. For this application section, I will merely choose 4 examples by the DiLeu for biomarker discovery. As we know that Alzheimer's disease is a progressive neurodegenerative disease. Over the past 20 years, the deaths from the AD have increased by the 141%, and by the 2050, the number of AD worldwide is projected to be around 600 million. There is no doubt that the AD brings a huge social and societal burden to the whole world. AD is featured by several neuropathological hallmarks, such as amyloid plaques, neurofibrillary tangles, and AD often has a very non-clinical process, which can up to the 20 years. This makes the AD detection face several challenges, such as its difficulty in early detection of AD symptoms, and lack of technique for the asymptomatic AD detection, and lack some both sensitivity and specific biomarker for the AD. We know that the accumulation of protein aggregate and altered protein structure are the common feature of the neurodegenerative disease. AD is a prominent example. Researchers have identified that the oligomeric and the aggregated state of tau in AD, and they found that the tau pathology progression is highly related with the AD stage. However, the method can be used for the large-scale analysis of protein conformational change in complex biological samples are still lacking. Given that the AD involves the misfolding of a low protein, we hypothesize that the analysis of protein structure could offer us new novel insight into the AD and potentially help us to give identification of new conformational biomarkers. To probe the protein structure change in complex biological sample, Professor Paula Picotti and her team proposed the limited protein analysis mass spectrometry. In this technology, the protein firstly undergo a short limited protein analysis under native condition, then followed by the fully tryptic digestion under the dilatory condition. This process will give us some informative fragments used to identify the protein region undergoing the structure change. Though the LIPS have many successful applications in several model systems such as drug-protein interaction, protein complex, and metabolite binding. However, only one sample in a single LC-MS can be analyzed. To enable high-throughput analysis in a single LC-MS raw, we propose to combine the DiLeu aspartic tag with LIPS. This combined approach, termed as the DiLeu LIPS, it can enable high-throughput structure quantitation in the highly complex biological sample. This is the workflow of the DiLeu LIPS. Briefly, it can be divided into the 3 parts. The first part is limited protein analysis. The second part is DiLeu peptide labeling. The third part is data acquisition, data analysis, and visualization. Before I introduce some results from the LIPS, I want to explain the 2 terms and general principle in the LIPS. The first terms I want to explain is the polytryptic peptide. Here, this kind of peptide indicates that shows the significant abundance change in the LIPS sample, and we can use that to identify protein region that undergo the structure change. The other term is a half-tryptic peptide. This kind of tryptic peptide is come from the internal cleavage of the fully tryptic peptide. Briefly, you can see this fully tryptic peptide is a longer peptide, while the half tryptic peptide is a short peptide. Because I think this two term is difficult to understand sometimes. The general principle in the lips is that we can theoretically we can use the fully tryptic peptide and half tryptic peptide to confirm the conformational conclusion in the lip experiment. Theoretically, a fully tryptic peptide and half tryptic peptide, they should display the inverse change in abundance in the lip sample, while the fully tryptic peptide, they show the comparable level in the trypsin group. The initial, so far, do we have any question? Okay, if no, I will continue. As the initial assessment, we used the myoglobin as a model system. Since the hollow myoglobin is a globular alpha heliofold which can convert into its apo form after the heme dissociation. This linked to the fully tryptic peptide in the hollow myoglobin is significantly higher than that in the apo hollow myoglobin, while its half tryptic peptide in the apo myoglobin is higher than that in the apo myoglobin. From this figure, we can find that the fully tryptic peptide showed the comparable level both in the hollow myoglobin and apo myoglobin. Based on the promising result from the above label-free lips quantitation, we further evaluated the accuracy and the dynamic range of DiLeu lips for the high-throughput quantitation for the structure protein change. From this figure, we can observe that the calculated DiLeu ratio matched well across with the theoretical ratio across the all channel, either in the 1:1 ratio and or in the 1:5:10 ratio. This result demonstrates that the DiLeu lips method can be used for the high throughput quantitation of protein structure change across a usable dynamic region. Next, we try to evaluate if this method can be transferred to the complex AD human serum sample. And from the result, we can find that some fully tryptic peptides have more than one lip cleavage site. Here, the green sequence indicates the fully tryptic peptide, while red color labeled residue indicates their lip cleavage site. From this bar chart, we can find that both these half-tryptic peptides from their fully tryptic peptide, both they showed the significant change between the AD and control group, and they also showed the upregulated level in the AD compared to that in the control. Here I want to mention that the conformal tryptic peptide includes both fully tryptic peptide and half-tryptic peptide. And from this figure, we can find that all these half-tryptic peptides were only detected in the lip sample, while the fully tryptic peptide were only detected in the lip trypsin group, and both they showed the comparable level. This result indicated that this protein did occur the structure change. Since the age and gender are regarded as the primary factor of the AD, though how exactly they influence the AD stability are still clear. Due to the small sample size and close range of age in our current study, we did not find any age-related protein conformational change between the AD and control group. However, we did observe these four proteins. They showed the significant change in female sample, while this protein they showed the significant change in the male sample between the AD and control. To discover valuable molecular target for the AD at the protein level, we validate the dysregulated protein both in the leaf group and the trips group. We found there have 4 common proteins that shared both the leaf group and trips group. And from this box plot, we can find that these 4 proteins both they showed the upregulated level in the AD compared to that in control. After we performed the geo-classification of the protein change, the protein analytic pattern in the AD and control sample, we found that most of this structure changed protein mainly involved in the cellular anatomical integrity, binding, catalytic activity, biological regulation, and metabolomical process. In particular, we found that the KGD pathway identified the complement and coagulate the top score pathway. With the 5 complement proteins, they changed the conformational was involved in this pathway, such as the C1R, C3, and C9. Both they showed the positive association with the AD pathogens. This finding is consistently well with our current large-scale study of the AD state biomarker. For instance, we pairwise comparison among the AD MCI and AD normal and MCI normal. Both they revealed that these structurally altered proteins are related to the complement and coagulation cascade pathway, Both they showed the strong association with the AD development. And we also found the consistent change in 3 fully tryptic peptide in complement C1R subunit complement protein. And from this bar plot, we can find that there have 2 fully tryptic peptide and there half tryptic peptide. Both they showed the reverse abundance change trade from the normal MCI to AD across. And especially we found that there have 2 fully tryptic peptides, both they located in the CUB2 domain, and both they showed the increased abundance from the normal MCI to the AD. This result indicated that this CUB2 domain became less protein analytic from the normal to the AD development. And this result further give us some clue that maybe the C1R could be a conformational biomarker. Additionally, we detected other complement proteins have a structure change across the normal MCI and AD, both in CSF and serum sample. The consistency of this result into an individual data set. The data set further highlights that the reliability of the DiLeu for biomarker discovery. In summary, in this part, we find that the DiLeu lips is an effective method which can be used the high throughput quantitation of structure change protein in the AD serum sample. And we also find several complement protein and structure change they have a positive association with the AD. And we also find this method could have a great potential for other biological system application. Do we have any questions so far? Okay, I will continue. In this application, we will use the DiLeu to investigate the molecular mechanism underlying the phenotype of the overexpression of the ATS1 and ATS2 in the development of autism spectrum disorder. Autism spectrum disorder is a chronic condition which involves several features such as communication difficulty and social interaction impairment. Since the neuron link a functional secretory pathway to maintain the synapse, while the ER isolation process is helpful for this pathway, and this pathway highly depends on these 2 enzymes. One is the NTS1 and another one is the ATS2. Since the ATS1 and ATS2 are linked to the ASD, and the 2 study the role of the ATS1 and ATS2 in ASD. This application DiLeu was used to investigate the molecular mechanism underlying the phenotype of the overexpression of ATS1 and ATS2 by using the knockout mouse model. After performing the quantitative proteomics both of the hippocampus and cortex, we found that the hippocampus seems to have much more response compared to the cortex. Firstly, we can find that the hippocampus showed much more pronounced and distinct protein in compared to the cortex. Secondly, we found that the distribution of altered protein was more extensive in hippocampus than that in the cortex. Thirdly, we found that from this Venn diagram, we discovered that the hippocampus have much more significant change of protein compared to the cortex, and the overlap of protein between these two groups was minimal, just 27. From this heat map, we can see that most of these shared proteins have different expression in the hippocampus and cortex. Finally, the key GG pathway analysis revealed some distinct category between the hippocampus and cortex. Moreover, we used the gene network plotted with the significant change protein in the cortex sample based on the gene-signal component function database. We found that several categories related to the mouse phenotypes such as the Schaefer-Calotary C1 synapse, neural synapse, and post-synaptic density and post-synaptic specialization. Together, this finding revealed that the DiLeu-based unbiased proteomics analysis not only demonstrated a common response to the overexpression of the ATS1 and ATS2, it also identified some potential biomarker for the autism spectrum disorder study. DiLeu also was being used in a study to investigate the protein dynamic in children with B-cell acute lymphoblastic leukemia undergoing the chemotherapy. And in this study, we totally identified the 63 proteins. They have a significant change trend across the all time point investigated. And hierarchical clustering analysis of this 33 protein revealed that the variation between the experimental group was greater than the variation within each biological group. And especially, we found that there have 4 proteins showed significant change during the different stage of chemical therapy. This suggests the potential of the early biomarker for the central nervous system toxicity caused by the chemical therapy treatment. Additionally, DiLeu was used to systematically explore the proteome and extracellular matrix alteration during the human pancreas development. This study not only confidently revealed previously reported pancreatic cancer biomarker, they also identified some core ECM protein, and the validation by the immunofluorescent study matched well with the DiLeu result. This further indicates the accuracy of the DiLeu for biomarker discovery. In summary, this section will demonstrate that the DiLeu method can provide reliable and accurate high-throughput quantitation not only for the PTM analysis, it also can be used for reliable biomarker discovery across many biological systems such as Alzheimer's disease, autism spectrum disorder, and pancreatic disease. There have so many applications can be needed to probe in the future. Finally, I want to mention that in the field of the biomarker, the process is typically divided into 3 phases, such as the discovery phase, verification phase, and validation phase. Currently, the DiLeu method just focused on the discovery phase to analyze a much smaller sample and identify the potential biomarker candidate. A verification phase and the validation phase is highly needed to further enhance the statistical power and confirm the biomarker candidate. Thanks for our advisor Lingjun Li give us this opportunity and organize this workshop, and thanks for our collaborator Luigi provide us some precious sample and model system, and thanks for the funding support, and thanks for the Wisconsin Autoimmune Disease Research Center provides sample to our biomarker discovery and PTM analysis. Thanks for our lab member, and thanks for your attention. I'm happy to take any questions.